This video is part 4 of a 4-part series on physical or engineering framework for understanding and computing food safety. This video summarizes the frameworks, and if you want to see the details, that can be seen in parts 2 and 3 of this 4-part series. So the approach to mechanistic safety prediction is basically treating bacteria as a species inside the food, or the chemical component like a carcinogen. So in this case, maybe acrylamide that forms during the frying process. Using acrylamide as a species, and so either bacteria or the chemical as a species, then we can readily formulate the various aspects that happens safety wise using the well-known transport equation for the species. Safety prediction or mechanistic and quantitative way of looking at safety combines two different physics. The first one is the bacterial physics. Again, because we want to do this in a mechanistic way, we think of the transport equation as providing the framework for the bacterial physics, and let's get into more details of that. So what is bacterial physics? It can be transport, okay, and transport in turn can be, let's say, migration or attachment. Now migration, bacterial migration can be passive with flow. So if it is passive with flow, then it becomes and it's a directed motion, it becomes part of the velocity in the transport equation. Or if it's passive and random, like Brownian motion, that becomes part of the diffusion term. Likewise, it can be active. So if it's active, like chemotactic, so it's a directed motion, it has a velocity, so then it becomes part of the velocity term. And motility, which is more random, then it will be part of the diffusion term. Then there is also attachment that has additional physics involved. Then after transport, the other possibility is, for example, growth. And growth can be included using primary or secondary models, and that's part of the source term. Likewise, inactivation, the same type of models can be included as part of the source term. Carcinogens can be treated in a very analogous way. So again, there can be just diffusion or movement with flow, or it can have reaction, meaning it's generation of more carcinogens. So both bacteria and carcinogen can be nicely treated in this transport framework, the details of which were discussed in the previous two videos. Now notice, when we're trying to do, let's say, the bacterial physics, so we need, let's say, how bacteria is growing or it's getting inactivated. That rate depends on temperature, and temperature can vary inside the food, okay? So we need that temperature information. Another thing is, let's say bacteria is being passively carried, and so we need the velocity. So the velocity of bacteria would be same as the velocity of the fluid that's carrying, which means I need to know how is the process physics, meaning what is the velocity of the food material. Because they depend on temperature, velocities, and so on at a location for the process, we need to combine the process physics, the food physics, with the bacterial physics to obtain the safety prediction for each scenario. Here is an example for carcinogen, let's say in meat cooking. So we're going to combine the food physics, the physics of how the hamburger, let's say, cooks, and the temperature varies inside, and if there are flows of liquid coming out of the meat and so on. So we do all of that, and then we combine that with the bacterial physics, which would include inactivation, for example, of microorganism. Or it can be, for carcinogens, it can be generation of the carcinogens. By combining the food physics that gives temperature and so on, and let's say the generation of carcinogens, we have a very comprehensive information that gives how much carcinogen is formed at each location and with time. So the framework for safety prediction combines the two physics. One is the food physics, and the other is either the bacterial physics or the carcinogen physics, or both. It is the combination that gives the safety prediction framework. So combining the two physics is the main idea. Here, the same framework is shown slightly differently with an added and a critical component at the end. So let's go through the details. First is we need microbial kinetics, that's kinetics of microbial growth or destruction, or carcinogen formation kinetics. Now that is local, meaning it depends on the temperature or other variables at that location. But temperatures can vary in a process, like it's shown here, that throughout the food, the temperatures are typically different, and they also change with time. So the rate of growth or the destruction kinetics are different depending on the local values. So it means to get the idea for the entire food material, we really need to combine the process physics with the microbial or carcinogen kinetics or microbial or carcinogen physics. By doing that, we will get microbial concentration everywhere in the food or carcinogen concentration everywhere inside the food. So that is the integrated process and local kinetic information. And after that, then we further process the data depending on what the context is. Our interest, for example, could be the location where we have the maximum amount when there is growth, or our interest could be the minimum point, or where we have the minimum amount of destruction. It could be a total amount, let's say, of carcinogen formation. So all of these are extracting critical information after we have combined the microbiological physics with the process physics. This is the added step that we are seeing in this slide. So what we are showing in this slide is the mechanistic framework for a food safety prediction in a very conceptual way. It can accommodate many different types of process or many different types of bacterial physics or carcinogen physics. The mechanistic framework that we just discussed can be nicely extended to what's known as risk calculations or food safety at a higher scale by combining the framework with additional information. So here I just want to mention the possibilities, not going into in any detail. So the mechanistic framework, for example, can provide a, let's say, reduction in bacteria, or in some other example could be the amount of growth in bacteria. We can combine that with initial number of bacteria, the initial load, or any recontamination. By doing that for a number of processes, so for example, it could be storage, transportation, cooking, and so on. By doing all of that, by doing this for every one of them, we can combine together and get the final number of bacteria or the final amount of carcinogen in a food serving. We then get consumption data. How much of this food is consumed by a person? We then bring in epidemiological data that says for a certain amount of ingested, certain amount consumed, what is a certain amount of bacteria or carcinogen consumed, what is the probability of illness for the person. We combine all this at the population scale, and that's what is referred to as risk. So risk would be the probability of illness for the entire population. It is important to see how the mechanistic framework that we have talked about so far eventually fits into this risk framework, and therefore can give a mechanistic description of food safety all the way up to the population scale. Details of such risk calculations are provided in separate module.